Today I'm talking through an AQA GCSE Biology Paper, Paper 2H from June 21. The nucleus of a cell contains DNA. Name structures inside the cell that contain DNA. Remember, these strands are known as chromosomes. Figure 1 shows part of a DNA molecule. Remember, these are the bases. And look at this pentose sugar here. That is, remember, deoxyribose in the case of DNA. Name the part of the DNA molecule labelled X. Again, that pentose sugar. You don't have to label it deoxyribose to get the mark. What type of substances are labelled A, C, G and T? In figure 1, these are the bases. So quite a lot of in-depth knowledge needed for DNA here. Figure 2 shows another section of DNA. For the substances you named in question 1.3 are not labelled in part Y of figure 2. Label each of these substances with the correct letter A, C, G or T and use information from other parts of figure 2 to help you. Okay, I think this question's a bit confusingly worded, but it just wants the name of these bases. Remember, the complementary base pairing rules state that A goes with T, C goes with G. So that's why these are the bases you need to label. What is happening to the DNA in part Z of the figure? Differentiation, no, that's all to do with cells specializing. Evolution, definitely not. Fertilization, when a sperm and egg fuse. Replication is the most sensible answer. A gene is a length of DNA. What type of substance does a gene code for? Remember, your definition of a gene is a section of DNA which codes for a protein. Most human cells contain 6 times 10 to the minus 12 grams of DNA. What mass of DNA will a human sperm cell contain? Well, sperm cells are haploid, which means they contain half the amount. So let's halve that number, which is why this is the answer. What is the name of the type of cell division that produces sperm cells? Well, sperm cells are examples of gametes. Remember, you need meiosis to produce gametes. Binary fission is all to do with bacteria replicating. A student investigated the effect of temperature on the decay of milk. Figure 3 shows the apparatus used. This is the method used. Set up the apparatus as shown in figure 3 with the milk at 20 degrees. Record the pH. Repeat with another batch of milk at 25. How could the student keep the milk at constant temperature for 5 days? Using a hot water bath. Give one variable the student should keep constant. Do not refer to temperature in your answer. Well, you shouldn't, because that's after all what we're changing. So anything sensible here, such as the volume of milk, type of milk, whether it's semi-skim, cow's milk, oat milk. Let's go for volume of milk. Table 1 shows the student's results for milk at 20 degrees. Complete figure 4. You want to label the x-axis, use a sensible scale, plot the data, draw a line of best fit. Let's go 0, 1, 2, 3, 4, 5. 0 was pH 6.7, 1, 6.7, 2 days 6.3, 3 days 5.3, 4 days 4.6, 5 days 4.4. I've labelled, I've got a scale, I've plotted. Let's draw a line of best fit. There's my curved line of best fit. The data you plotted in question 2.3 were obtained at 20 degrees. Sketch line on figure 4 to show what you'd expect at 25 degrees and label the line. You'd expect the pH to just drop faster because you've got a higher temperature. Remember, it would start at the same pH. Human body temperature is controlled within very narrow limits. Scientists investigated the effect of drinking ice-cold water on internal body temperature, the rate of sweating, This is the method used. Sit a person inside a room kept at constant temperature of 25 degrees. Measure the person's internal body temperature near the brain. Measure the person's rate of sweating. After 20 minutes, give the person 500 centimeters cubed of ice cold water to drink. Continue to measure the person's internal body temperature and sweating for a further 50 minutes. Give the reason why the person should not move during the investigation, because if you're moving, you might be sweating a bit more. Figure 5 and 6 show the scientists' results. So here's the internal body temperature dipping when they've had that cold drink, and then it rises again. And the rate of sweating decreases as well when they've had a drink. What is this person's normal internal body temperature? It's this value here, 37.4. The results show that when the ice cold water was drunk, the temperature near the brain decreased. Explain why the temperature near the brain decreased. So we know it's the blood running through the brain that's gotten colder. Why would it have gotten colder? Because that blood would have been cooled either at the stomach or at the mouth or both. And then that blood flows off to the brain. The thermoregulatory centre in the brain responds to the decrease in temperature. How does the thermoregulatory centre send information to sweat glands in the skin? Via nerves. The rate of sweating changes between 24 minutes and 36 minutes. Explain how this change helps to maintain the body's normal body temperature. 24, 36. Well, you can see here that less sweating takes place, meaning that less heat is lost. During exercise, the skin appears red. What causes the skin to appear red? This is all to do with vasodilation. Dilation of blood vessels means the same thing. Decay occurs in a compost heap. Figure 7 shows a compost heap. Describe how microorganisms in the layers of soil help to recycle chemicals in the dead plants, how chemicals are used again by living plants. So the microorganisms decay the dead plant, releasing things like nitrates. Then how are these nitrates used by living plants? Well, nitrates are absorbed by active transport into the root hair cell. They really want a lot of detail as to how this absorption takes place. Where they're used to make amino acids needed for proteins in the plant. And then because these microorganisms are living, remember they're going to carry out aerobic respiration. And why is that a good thing from a plant growth point of view? Because it releases carbon dioxide, which is absorbed into the leaf of the plant via the stomata, those small pores in the leaf, and the gas is used in photosynthesis to make glucose. Let's just check how many marking points we have. Nitrates as a marking point, active transport, amino acids, respiration being mentioned, production of carbon dioxide, the stomata used in photosynthesis to make glucose. So 1, 2, 3, 4, 5, 6, 7, 8 different points. The growth of daisy plants on a lawn is affected by biotic factors and abiotic factors, so living versus non-living factors. Table 2 shows 6 factors. Tick one box in each row to show whether the factor is biotic or abiotic. So nitrates in the soil, non-living. Rabbits eating the plants, a living factor. Shading by a building, abiotic. Soil pH, abiotic. Temperature, abiotic. People are alive, so biotic. Figure 8 shows a plan of a garden. A student estimates the number of daisy plants growing on the lawn. The student places a quadrat at 10 different positions on the lawn. The quadrat measures 50 centimetres by 50 centimetres, and the student counts the number of daisies in each quadrat. How should the student decide where to place the quadrat? Give a reason for your answer. Remember, placing quadrats is all about placing them randomly in order to avoid bias. So how can we do that? The best way is to kind of create a grid system with coordinates and use a random number generator to help you place that quadrat. The mean number of daisy plants in each quadrat is 6. Calculate the number of daisy plants on the lawn. Give your answer to 3 sig fig. So we need to work out how many quadrats we would need to place on this lawn. So first of all, work out the area of the lawn. It's a triangle, so we do half times the length, which is 16, times the height, which is 80 meters squared. Let's remove the paved area because obviously there's no lawn there. That's just 6 times 3, which is 18. So take away that 18 to get 62 meters squared. And then each quadrat measures 0.5 meters by 0.5 meters, which is 0.25 meters squared. So we now need to divide the 62 square meters by 0.25 to get 248. And then we were told that each one contained 6 daisies as a mean. So just multiply those two together, which to 3 significant figures is this number here. Make sure you put all your working. I just showed it up here so you could see how I was arriving at my answer, but really this should all be down here. Using the mean from this investigation to calculate the number of daisy plants on the lawn may not be accurate. Give 2 reasons why. First of all, they haven't placed enough quadrats. And then the ones they did place might not be representative of the lawn because some of the lawn might be in shade. Some of it may be affected by rabbits eating the daisies, so yeah. Reflex actions are coordinated by the nervous system. What is meant by the term reflex action? It's a rapid, automatic response to a stimulus. Learn this definition. A woman's hand accidentally touches a hot object. The woman moves her hand away. Describe how the woman's nervous system coordinates the reflex action. This is a gift, 6 marks. This is an answer you just need to learn. It's a great way of guaranteeing 6 marks. Remember, my revision guide is full of these sorts of answers. So we start by saying the receptor in the woman's hand detects the stimulus, which in this case is the hot object. What's the next step? Well, an electrical impulse is sent along the sensory neuron to the relay neuron, and then lastly, motor neuron. Then the effector brings about the response, which in this case will be a muscle contracting to draw her hand away from the hot object. And then remember, the gap between 2 neurons is called a synapse, where a neurotransmitter diffuses across. The endocrine system coordinates many internal functions of the body. Give 3 ways coordination by the endocrine system is different from coordination by the nervous system. Endocrine system should be screaming hormones at you, whereas nervous system is all to do with nerves or neurons transmitting electrical impulses. Let's make a full comparison. Endocrine system uses hormones. Nervous system uses electrical impulses. The endocrine system is slow. Nervous system is fast. Endocrine system response is long-lasting. Nervous system response is short-lasting. Describe how hormones control the menstrual cycle. We want to talk about FSH here, LH, estrogen, and progesterone. So let's start with FSH. FSH, follicle stimulating hormone, as the name suggests, stimulates the follicle, which is otherwise known as the egg. So the FSH matures the egg in the ovary. LH causes ovulation, which remember is release of the egg from the ovary. Estrogen inhibits FSH and LH production, whilst also thickening the uterus lining. That's important because, after all, if you're waiting for that egg to land at the uterus lining, you don't want to be maturing or releasing anymore. And then progesterone is important because, again, it inhibits LH and FSH production whilst maintaining the uterus lining, because we need to make sure that fertilized egg stays implanted. Food web contains several food chains. Figure 9 shows a food web. The animals in Figure 9 get their energy by eating other organisms. Describe how algae get their energy. Remember, they photosynthesize and absorb sunlight energy in order to make glucose. Name one primary consumer in Figure 9. So that's something which eats the algae, so that could be krill or copepod or even the herring, so any of these arrows here. Name one producer in Figure 9. So that's something which photosynthesizes, such as algae. The different food chains in Figure 9 have different numbers of organisms. Complete Figure 10 to show food chain with 5 organisms, including the human. So we know we need to start with algae. We need to end with human. So we need 3 organisms between them. 1, 2, 3. Because the energy flows algae to krill, to squid, to mackerel, to human. Figure 9 shows that mackerel eat krill and squid. The biomass of mackerel is much less than the combined biomass of krill and squid. Give one reason for this. One reason for this is that mackerel can't digest all parts of the krill and squid. Give 2 other reasons. So where's energy lost? Well, the mackerel excretes urea. And remember, other energy losses include respiration. You can't say stuff like keeping warm here because it's a fish rather than a mammal. Or muscle contraction. I mean, you could, but I think let's just say it respires. Also, you can say that there are some parts which aren't eaten. Figure 11 shows how the biomass of adult herring in the North Sea has changed between 1915 and 2010. Calculate the percentage decrease in the biomass between 1960 and 77. Give your answer to the nearest whole number. So we need to look up here. Make sure you're picking the right years there. So percentage decrease is given by the equation change over original times by 100. So let's read this off. That's 2.3. That's 0.1. So the difference between those numbers is 2.2. The original was 2.3. Times it by 100. So our percentage decrease is 95.65. To the nearest whole number is 96% decrease. That's quite worrying. Too many herring were caught by fishermen between 1960 and 1977. Herring can live for up to 12 years and reproduce aged 3 to 4. Laws have been introduced to help conserve herring, such as banning fishing, control of mesh size, fishing quotas, herring fishing banned in breeding grounds during the breeding system. So this is obviously to just increase the numbers. Allows small herring to escape. Fishing quotas limits the numbers caught. And then it allows them to breed. This ban on fishing during breeding season. Figure 12 shows how a minimum mesh size helps to conserve herring. Oh wow, look, they've drawn it. Look, the little baby fish can escape. Figure 11 is repeated below. Evaluate the effect of these laws on the conservation of herring stocks. Use data from figure 11 and information from figure 12 in your answer. So evaluate means, has all these measures, have they worked? And give evidence for and against. Let's start by what has worked. The laws have worked because let's look between 1977 to 1981. We can see that the biomass of adult herring has increased. Let's start by saying that. Let's quote numbers. So from 0.1 to 0.5. From 1984 to the present day. So from here onwards, yeah, we can see again increased biomass. So introducing mesh nets has increased biomass. As small fish are able to escape and reproduce. And then this whole, from 1997, 1998, fish quotas and herring fishing being banned during the breeding season. Again, we can see that increase. So fish quotas and ban on fishing during the breeding season has also increased herring biomass. Well, what about evidence against? So the most straightforward thing against it is remember that correlation does not always mean causation. So yeah, so what? You've stopped fishing, you've introduced these mesh sizes, the quotas, the lack of hunting during the breeding season. We have no real proof that that is the reason why the biomass has increased. I mean, of course, it's highly probable. So points against this conclusion include the fact that correlation, please always use this point, does not mean causation. Because all these laws are superimposed on top of each other in terms of they're all overlapping, it's really hard to tell which one had the biggest effect. And one thing that's really horrible is the way that quotas work is it means that a lot of dead fish are being thrown back into the sea. So again, our data here isn't going to really hold true. Sickle cell anemia is an inherited condition affecting red blood cells. It is caused by a mutation in the gene for hemoglobin. Hemoglobin is a red pigment found in red blood cells. A person who is homozygous for the normal hemoglobin gene produces normal red blood cells. So let's just write what that means. Homozygous meaning the same alleles. So you're going to get normal red blood cells in this situation. A person who is homozygous for the mutated allele HS produces red blood cells with abnormal hemoglobin and ones that have a sickle shape, meaning that the person becomes ill. So there's the anemia phenotype. A person who is heterozygous, which means one of each, has both normal and abnormal hemoglobin, has a sickle cell trait, is generally healthy. So they're carriers. Give the reason why a mutation in the gene coding for hemoglobin could be harmful, because less oxygen is transported, meaning less respiration takes place. Figure 13 shows some red blood cells from the blood of a person with the sickle cell trait. Calculate the proportion of cells that have an altered shape. So let's highlight them 1, 2, 3, 4 over the total number of cells 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, which is 0.24 to two significant figures. Figure 14 shows the inheritance of sickle cell anemia in one family. Here's a key. Persons 8 and 9 are expecting a second child. Determine the probability that the child will be a girl with sickle cell trait. You should draw a Punnett square, identify the phenotype of each offspring, and use these symbols. So let's identify persons 8 and 9. So the unaffected male, remember we said, was HAHA. Person 9 is the female with the sickle cell trait, which remember is HAHS. So let's lay it all out. Mum, dad. I always lay out my answers like this. It's really good practice. So we're not setting up our Punnett square quite yet. We're just getting ourselves in the right position. So the mum has the sickle cell trait. Dad is healthy. The genotype then we've said was this. So the mum's eggs are either HA or HS. The dad's sperm are all HA. Let's draw a Punnett square. Mum, dad. So here's their potential offspring genotypes. Leave room to put their phenotypes. So this first offspring will be normal or healthy. This one will have the trait for sickle cell, normal. This one will have the trait. The question was to do with the sickle cell trait. So 0.5 chance or probability of having the trait. But remember, we needed it to be a girl. So there's a 0.5 chance of the child being a girl. So you need to multiply those two numbers together to make sure you have both a girl with the trait to get 0.25 as your answer. Just double check you've done everything. I've done the Punnett square. Here are the phenotypes. And they've been attached to the genotypes. And here's my final answer, and that is worth a whopping 5 marks. Without medical treatment, people with sickle cell anemia are frequently ill and have reduced life expectancy. The malarial parasite cannot live in red blood cells of a person who has the HS allele. A scientist stated, "It is an advantage for people to have the HS allele in countries where malaria occurs." Evaluate the scientist's statement. Yes, so again, for and against. I agree because the HS allele stops infection by the malarial parasite. HAHS people neither have sickle cell anemia or malaria. However, their offspring may inherit, because if their parents have HS, then they may get 2 copies of the HS, which would mean that they have sickle cell anemia and therefore, without treatment, have reduced life expectancy and a great deal of suffering. The Galapagos Islands are located in the Pacific Ocean. Several species of birds called finches live on the Galapagos Islands. These finches are very similar to each other. Figure 15 shows 2 modern species of Galapagos finch and their classification. We have the medium ground finch and the small ground finch. Complete figure 15 to give the names of the missing classification groups. So we use the mnemonic: king, prawn, curry, or fat, greasy. Sausages, you may have your own, but what that means is we're missing prawn, which stands for phylum, or, which stands for order, fat, which is family, and sausages, which is species. Give the binomial name for the medium ground finch. Looking at this column, we need the genus and the species name, so it's Geospeza fortis. In each species of finch, there is variation in beak depth. Figure 17 shows the relationship between the beak depth of the parent birds and the beak depth of their offspring. Give evidence in Figure 17 that shows that beak depth is an inherited characteristic. You can kind of see this relationship up here, which is that when the parents have deeper beaks, so do their offspring. When they have shallower beaks, so do their offspring. So offspring beak depths are similar to their parents. Scientists suggested that more than one gene controls beak depth. Give evidence from Figure 17 to support the scientist's suggestion. Well, if you look here at this mean beak depth of the parent, well, there's a lot of different depth beaks from the offspring's point of view. So parents of a given beak depth produce offspring with several beak depths. Figure 18 is a map of the Galapagos Islands. On Isabella Island, the medium ground finch and the small ground finch are found. On Daphne Island, only the medium ground finch is found. Cosman Island, only small ground finch is found. Figure 19 shows how beak depth of each species varies on each island. So here's Isabella Island, and as you'd expect, we have both the small and medium ground finch. Daphne Island, we've just got the medium finch. Crossman Island, we just have the small ground finch. The medium ground finch and the small ground finch both feed on seeds. The size of seeds eaten by each bird depends on the depth of the bird's beak. The range of beak depth of medium ground finches on Isabella Island is different from the range on the Daphne Island. Explain what might have caused this difference. So remember, on the top island, Isabella Island, we've got a good selection of both small and medium ground finches, which means there must be quite a variety of seed sizes here. And remember, those beak depths all come about due to genes, so there'll be a whole range of genes which are providing a whole range of beak sizes that are suitable for both small and large seeds. However, if you think about Daphne Island, where there's just the medium ground finches, this time you're going to find larger seeds, meaning that you want finches with larger beaks, and that these ones are more likely to survive, they're more likely to reproduce and pass on that gene for large beaks to their offspring. Let's start by talking about Daphne Island. Figure 19 shows that two species of finch live on Isabella Island. One species lives on Daphne, only one species lives on Crossman. Suggests why both species of finch are able to live on Isabella Island. Mainly because it's such a large island, and therefore there's enough seed for all the different bird species and all their different beak depths.